Welcome everyone to today's Technicals webinar about simulating cytokine-related therapeutic protein DDIs using the new TP-Modulator function that was implemented in version 23. I'm Felix Stader, part of the biologics team, and it's my pleasure to host this technical webinar today. Some general comments before we start. Please make sure you're all muted so we don't have any disturbance during the session. If you wish to ask any questions, we have a dedicated Q&A session at the end of the presentation. So please type in your questions into the chat functionality, or in the end, use your raise hand function to be unmuted and ask your question. The webinar will be recorded and will be available on YouTube shortly. So now it's my pleasure to introduce my colleague Jian Pan, who is part of the translational DMPK team, and who led the effort in developing the new model, the new TP-Modulator modality within the Simcyp simulator. Since joining in 2017, she worked on all kinds of small and large molecules, also on pediatrics and pregnancy models. What we will cover today is the introduction into cytokine-related DDIs, and also look at how we can use the Simcyp simulator to simulate 3 different kinds of scenarios, and also see how we are setting up the screens. So Jian, the floor is yours.
Thank you, Felix, and hello, everyone. So now I'd like to start with some background introduction. So in June of last year, FDA released the guidance on the DDI assessment for therapeutic protein. And last month, ICH released the final version of M12 drug interaction. But both FDA and ICH recommend that when evaluating the TP-DDI potential, the sponsor should consider the DDI mechanism, disease condition, clearance of therapeutic protein, as well as a co-medication in patient populations. The FDA also highlights the utility of PBPK model in providing mechanistic understanding of the DDI potential for therapeutic protein. The pro-inflammatory cytokine level can be elevated in disease conditions, post-surgery, or the drug treatment such as bispecific antibody or even small molecule treatment. So the increased cytokine level can suppress the CYP enzyme and therefore decrease the metabolism of small molecule drugs that are CYP substrate, while the CYP modulator can remove these suppression effects on CYPs and restore the CYP activity. Subsequently, you will see the increased drug clearance and decreased drug exposure. In version 23, we have introduced the TP-Modulator module to mechanistically model the cytokine-related TP-DDI. So in this new module, have connected the Simcyp biologics model with the Simcyp small molecule model, where the CYP suppression is driven by the concentration of the cytokine receptor complex. So we account for the interaction among the cytokine modulator, cytokine receptors in the specific cases. So we have introduced a new compound slot named the TP-Modulator, which is used to model either cytokine or the cytokine modulator. So this slot is specifically designed to function as an inhibitor with interacting with the small molecule drugs. So it is compatible exclusively with compound type as a large molecule. There are various dosing rules can be used, and the model is available for all the adult populations except pregnancy, where the biologics model is not yet implemented. We have not extended this module for pediatrics yet. There are 2 specific screens which exclusively to the TP-Modulator slot. They are the interaction screen and the target-mediated interaction TMI screen. And you can find this tutorial and the workspaces that will guide on the model setup and parameter entering for various clinical relevant scenario, and we hope it will help you become familiar with this new module. And to access to these materials, I just switch to simulator. So we open up the main simulator from the resources hyperlinked file. If you click that, it will direct you to this folder specifically for the TP-DDI, or we call it the DBI module. The tutorial is here, and we also have workspaces related to different clinical relevant scenario that you could set up. So it's just pre-set up for you, and you can use them directly, input the meaningful values, and run the simulation. I will go through each of them with you shortly, but before that, I will briefly show you what this tutorial look like. So if you open that version 23 DBI module examples, you will see this tutorial is for the version 23 setup. And the content-wise, it's explain what this slot for, what module can be used, what was included, and importantly is the application. And within this application, we divided the clinical relevant cases, which could simulate it into 3 different scenarios. And for each scenario, we have prepared the workspace for you, as you can see in other folders for each of them using different modules. And the detailed explanation about how to set up them are all shown in this tutorial. And in today's webinar, I'm going to go through each of them. You might wonder what we mean by each scenario, and I have a slide for you so we could all have a look at them. So essentially, this new module can assess the TP-DDI liability across several different clinical relevant scenarios. And we have simply divided them into 3 categories as illustrated in this slide. We're going to go through one by one. So in scenario 1, the cytokine or the cytokine products is characterized as a large molecule that interacts with small molecule drugs by modulating Cyp expression. So this setting can be utilized to explore the impact of chronic elevated cytokine levels in patients under different disease conditions, such as inflammatory disease, cancer, or COVID. Or it can also be used to assess the transiently elevated cytokine levels or dynamically fluctuating cytokine levels post-surgery or post-treatment with therapeutic protein or even small molecule drugs. So that's what you can use this scenario to model. For instance, as we're showing here, this plot here, when the IL-6 level is increased, the Cyp enzyme activity is reduced and leading to a decrease of the Cyp substrate clearance and increase of exposure. As you can see, its concentration time profiles for the small molecule drug with a increase of IL-6 level, you will see the increased exposure indicate decrease of this clearance. And with that, I can switch back to the simulator and showing you this scenario 1. So this where you could just open from that linked DBI workspace folder and select the first scenario 1. Then you're going to load it in the simulator. So here, this is the TP-Modulator. In this example, we model the IL-6. So all these things are all related, all these input are related to IL-6. And the particular things you might be interested in, the first one is this TMDD part, because we said that within our model, the driving force or the driving concentration to suppress the Cyp enzyme expression and activity is a cytokine receptor complex concentration. So we use this TMDD to describe the cytokine binding or interaction with cytokine receptor and the form of the cytokine complex. So this is an important thing. You need to set it up, otherwise you will not see the interaction. So for the IL-6, we modeled here the interaction with membrane binder receptors. You will notice that in the healthy volunteer population target screen, we have already having this IL-6 membrane binder receptor level in the liver and the gut implemented into the simulator, so you can use them straight away, and you will see them from the drop down. You can click them. And important thing here, when you define the target, you need to select the right location for them, otherwise they can have mismatch. And then in the interaction part, you defined the interactions. So within TMDD screen, you get multiple models to describe the TMDD. Here we use the QE model, and those are data from the in vitro. So in this case, you don't need the TMI screen. That will be used in the scenario 2 and 3. What you need is to set up the TMDD screen and also the interaction screen. For the suppression, you just input the Inmax and IC50 or both of them, or the slope model. If you're familiar with a small molecule interaction model, these are the same as that. So we already have some values in this example that's also derived from the in vitro suppression study for all these CYPs that's mediated by the IL-6. Another thing I want to show you is the output screen. So of course, there are several things you can output. I just take several of them that's more related than you might be interested in. So in the PK profile session, the plasma concentration profile output can output the small molecule profile with and without interaction of IL-6, and also the IL-6 profile or this TP-Modulator profile. So you will see all of them on the same output sheet. And the AUC under the PK profile can output the AUC of small molecule drugs. And for the TP-Modulator AUC output, you need to select under the TP-Modulator session. Another thing you might interested in is the enzyme profile as the cytokine suppress or modulate the CYP expression. You will see the difference of this profile. So that might be another thing you want to look at. If you click the hyperlink, it will lead you to the different CYP selection. Here I selected 3, 4 just to show you the results. And also the TMDD that's related to the interaction and the driving force. So I can select all these, but there are, of course, other things you could look into. Onto the trial design screen, those are the same as the similar. Well, there is nothing change on the trial design screen. The only difference here is the dosing. You need to define the dosing for the TP modulator. In this case, we're trying to model the chronic elevated IL-6 level. So I use the IV infusion dosing for a period of long time until it reached the steady state. So here, just using this session to define the dosing. And for the small molecule, you can set up on other session here. You can run the DDI along with multiple small molecules. You could have a substrate, inhibitor 1, 2, 3, and the primary inhibitors. So I already ran the simulation and I can show you the results. So we can all look at how the output look like. The concentration time profile screen, we have this for small molecule with and without the interaction with IL-6, and the IL-6 profile, they are all on the same screen. And the interaction, the AUC sheets, you might interested in look at the AUC and the Cmax ratio. As I mentioned about the enzyme activity or the enzyme abundance changes, you can see with the increase of the IL-6, the suppression effects on the CYP enzyme. And also we have output the TMDD parameters that you define, and they're just showing you individual parameters and also the profiles. So that's including the free drug, complex, drug target complex, and free target, target occupancy, and the total target and the total drugs. So those are all in an output if you select the TMDD profiles. So that's pretty much about scenario 1. And then we can move to the second scenario. So in the scenario 2, basically that's investigate the TP-DDI between a cytokine receptor antagonist and small molecule drugs. So here the cytokine receptor antagonist is characterized as large molecule, and we will use the TP-Modulator slot to modulate. And its therapeutic target is the cytokine receptor. So the TMDD screen going to be used to model the interaction between the therapeutic protein and the cytokine receptor. And in this case, and also scenarios 3, you will also need a TMI screen where they in there, you could define the endogenous cytokine receptor interaction. So basically, the cytokine receptor antagonist prevents the cytokine from binding to its receptor, then will reduce the level of cytokine receptor complex, and subsequently release the suppression of CYP enzyme. As a result, you will see the CYP activity restored and the decrease of the drug exposure, increase of its clearance, as you see from this plot here. When you dose the tocilizumab, which is the cytokine receptor antagonist, you will see the small molecule drug exposure decrease where there's CYP activity restored. And then let's just move back to scenario 2. So there are 2 workspace for scenario 2 and 2 for scenario 3. That would just be the only difference is the TMI model and TMDD model difference. So there are 2 options for TMI model, either full or QE model. So I'm gonna open one of them for this, and then we could look into the screen and all the design. So here, we model using the TP-Modulator slot to model the cytokine receptor antagonist. And then you have TMDD screen to describe this drug interaction with the, for example, IL-6 receptor. And then on the TMI screen is where you define this endogenous cytokine and cytokine receptor interaction. So you need to define the both screen and also the interaction screen that's defined the interaction parameter, which is driven, the interaction going to be driven by the cytokine receptor complex concentration. As you see from TMI screen, we need the 3 input, the cytokine and also the cytokine receptor. All these you can define from the population target screen. And here we add in the IL-6 in this case, and you might want to change it in a different level to reflect the baseline concentration in different individual under the different disease conditions. Okay, and for the output wise, when you have the TMI model, you get the extra options where you can output the TMI parameter and the TMI profiles. Other than that, other things are similar as the scenario to all these screens. Again, the TP-Modulator dosing, you could define from here. And then I got one example where we have looked at the tocilizumab interaction with simvastatin. So you can see with the treatment of the tocilizumab, you see the restore of the small molecule simvastatin activity that's reduced exposure of this, as you can see from this dashed line. So this is concentration time profile screen. You got TP-Modulator profile and the small molecule profile with and without interaction. In the AUC screen, you could look at the interaction magnitude, AUC Cmax ratio. Also for the 3A4 interaction, because it's restored activity. And at the beginning or on the baseline, it's not start from 100. That was because from the baseline, you get the IL-6 and IL-6 receptor endogenous level, and that will set up the initial suppression level. So the CYP activity or bindings is suppressed at the beginning, and before you dose the drug and when you have the modulator dose, you're gonna restore it. As you can see, it's gradually increased, and with the elimination of the drug, it could just return back. So that's a profile you might expect it. On the new opposite for the TMI profile or and also TMI parameters. And so the TMI model is to define the cytokine receptor interaction. So you keep having this free cytokine complex, cytokine receptor complex, receptor, and the total level of this cytokine and the cytokine receptor. Those all reported here. Apart from that, the TMDD screen and outputs giving you the drug and the drug target interactions. So that's for the scenario 2. I'm gonna briefly go to scenario 3, which more or less the same as in terms of the setting, but the only difference here is the target. So at the in scenario 3, the TP-Modulator is an anti-cytokine antibody, so you're gonna target the cytokine rather than the cytokine receptor. If we come to the scenario 3, showing you the difference. So the difference mainly is on the TMDD screen. If you remember, in the scenario 2, the target is the receptor, and here you need to define the cytokine as a target and select a plasma here. And the TMI screen gonna be the same because that just describes the endogenous interaction between cytokine and cytokine receptor. So I think that's all I want to show for now, and I'm happy to take any questions. Thank you.
Thank you very much for this nice and informative presentation. Just to remember, if you have any question, please type it into the chat or use the raise hand functionality. So we have a question from Mary, which I will read. The clearance of interleukin-6 in this file is quite fast. Was it based on literature data? And how would you suggest to adjust the profile to match the observed data?
Thank you very much about the question. So for the IL-6 model, there are literature data where IL-6 is dosed in the adults, and then they have this concentration time profile. That's where we fit to the profile to optimize the clearance value too. So when you have, well, if you look at the different cytokines, that could be the way to do that, to just optimize the clearance value to pick up the concentration time profile we observed. I hope that answers your question.
Yes, thank you. I think it's kind of related to the fact that the infusion's not maybe directly related to the amount of IL-6 is being produced over time relative to the clearance. So you're suggesting then just adjust the clearance value, and that kind of will, versus like the amount that we're infusing and the time we're infusing, like how are we balancing those two so that we can kind of get the profile correct?
So, and for IL-6, the observed data is where they give in the IV infusion, and at the end of the infusion, they start sampling. So you will see this decreased profile, the concentration profile, not just to reach the steady state, it's after that. So use this, you could get the clearance and to fit to that. And there are multiple different dose levels, so you could get confidence that that's your model.
Yeah, but in our scenario, we're not actually dosing IL-6. The IL-6 is being produced endogenously due to the TP modulator. So that's why I was asking.
Yeah, that's fine, as we already have this module there. The IL-6 module is sort of like verified, and we're preparing the manuscript for that. So you could just use our module for that. And if you look at the TP modulator, it could actually when you go to the, so if you're using, I suppose you're looking at scenario 2 and 3, if you're looking at the TP modulator.
It's actually scenario 1.
Okay, so if just scenario
the
1,
the IL-6 level sync.
okay, I think if just the scenario 1, you could use our scenario 1 workspace directly or IL-6 file directly. That's fine.
Okay, thank you.
Okay, and then we have another question from Tanvi Desai, again regarding the interleukin-6 model. So can you go over the experimental data used in this model that is specific to the cytokine of interest? So which I guess this is which
scenario 1.
I guess this is which experimental data you need for a cytokine.
Okay, so let me just take IL-6 as an example. So for us to develop this IL-6 large molecule file, we're very likely to we're very lucky really to get this clinical data where they dosed the IL-6 intravenously and measure having this concentration time profile for that. So that help us to getting the clearance value right. And then what else you need, maybe I could just open scenario 1, is you need, as I say, you need to define this the interaction, the cytokine receptor interaction through the TMDD screen. So for the IL-6 membrane binder receptor, we go through the literature, and this actually derived from the cell-based assay. So there, if I remember correctly, they from the HapG2 and the KCO cells, and we do some extra calculation to derive this membrane abundance for the IL-6 receptors in the liver and the gut. So you will need these and to define them on the target screen, and also the degradation. Those we also get this from the in vitro study from the literature. And on here, the IL-6 or cytokine part, you will need this cytokine receptor interaction, the binding association rate constants. Those things you could use the QE model where you get this Kg as input, or when you have the K on K off, you could use the 14DD. For the internalization, in our model, we just assume they're the same as the turnover of the receptor, as they're just the limited of the data there. And when we get all these, another thing you will need is on this interaction screen. So those are from the in vitro data where they measure the IL-6 suppression on the human primary hepatocytes, and they derive this NC50 and the Inmax value. That's where we're putting. So basically, most of the things, they all from the in vitro, but we did optimize the clearance value to pick up the exposure of IL-6 in people. And then you can verify your model further through the DDI studies. I hope that might be helpful.
Yeah, thanks, Fran. That was really useful.
Okay, is there any last question? If this is not the case, then thank you very much for your presentation. Really informative to see how to set up the TP modulator in the workspaces. So can you please show the last slide?
Yeah.
So the The next technical webinar will be on the 29th of August about the dynamic pH-dependent DDI modeling and will be presented by Jan Yip, who is part of the absorption team. So if you're interested in that, please register on our website. Please also look for the other webinar series that we have. Thank you all for your participation. Have a great day and goodbye.